So, in 45 minutes, maybe, I am going to attempt to give you an overview of everything that you need to know that's important about aging, health, cancer, and nutrition. If I left something out. Previously, when I have talked or written, invariably somebody will say, you prove it, because I always tend to say things that are a bit contradictory to prevailing thought. Of course, you can't prove anything. All you can do is present evidence. And so I will be presenting a lot of evidence, and the only way to satisfy that requirement is to cite some studies. So my preference is just to talk, but I will be giving you lots of studies and just reading from them, and I'm going to go through them really quickly, as fast as I can, to give you just an overview, a taste of what they're talking about, to back up what I will be saying. And we've heard a lot about Paleolithic nutrition. The idea being that Paleo man knew better than we do how to be healthy, and that we can learn from how Paleo man lived and derive perhaps proper diet if we ever really knew what they ate. I'm going to maintain that that's insufficient for a number of reasons. Number one, it's assuming that nature cares about us being healthy in the first place. And that's very debatable, which we'll find out. Secondly, Paleo man, as far as the time frame, is a drop in the bucket. 2 million years ago? That's nothing. Really, the rules of life and health were laid down shortly after life began. And certainly from the transition between unicellular organisms floating around the oceans into multicellularity, when we had nutrient sensors and communication and division of labor, that is where the rules of life were laid down, and everything subsequently has built upon those shoulders. So we're going to start from there. We're going to start from when the oceans were filled with so-called protobacteria, the so-called Proterozoic eon that was from about 2.5 billion years ago to perhaps 500 million years ago. And that's when the transition occurred from unicellular to multicellularity. Ultimately, some of these protobacteria turned into cyanobacteria. Photosynthesis evolved. Cyanobacteria started filling the oceans and started filling the atmosphere with oxygen. Prior to this time, there was no oxygen in the atmosphere, or virtually none. Any little bit of oxygen would have been gobbled up by iron minerals that would have oxidized and sucked any oxygen from the atmosphere. So the atmosphere was anoxic prior to this time. So we think maybe that's a good thing that all of a sudden our atmosphere started filling with oxygen, but not such a good thing. That's considered and called the Great Oxygenation Event. Occurred a little over 2 billion years ago because life wasn't prepared for it. I mean, now we have antioxidants and things like that. We have mitochondria that uses the oxygen, but back then there was nothing. And it's estimated that at least 90% of all life was wiped out by oxygen. I mean, oxygen burns. And we talk about a little bit about oxidizing now, but at that time, it was mass extinction. Until one of the little protobacteria learned how to cope. That was the ancestor of our mitochondria. And they learned how to take that oxygen, and rather than have it burn themselves, they basically fed the oxygen, a sacrificial lamb, which was food, other hydrocarbons that the oxygen burned. And not only did it save the mitochondria by deflecting the oxygen away from itself, but it learned that there was a lot of energy to be derived. And so that was the beginning of respiration. Prior to that time, glycolysis was the name of the game. Glucose was everywhere. And glucose was being used as a fuel, but you can only make a little bit of ATP, a couple molecules per glucose. With respiration, you can make a whole ton more, 36 molecules for the same molecule of glucose. So you could end up with a whole lot more energy. So not only did it save the mitochondria from getting oxidized, but you could generate a lot more fuel, and that fuel was necessary then to fuel the evolution of multicellularity. And as the glucose became gobbled up, became more scarce, it behooved life to use energy more frugally. Shower with a friend, huddle together also from all the oxygen. Life learned to colonialize, to help protect it from being oxidized, but one had to pay a price for this colonialization and multicellularity because there had to be a division of labor, because some of the cells were on the outside, some were on the inside. The outside was exposed to more oxygen, but also more fuel availability. The inside was protected, but didn't have any food. And as life evolved into multicellularity, you had a division of labor, and not the least of which was a division between the so-called soma, which is the body, pretty much everything, and the gametes, which are essentially the future generations. So the soma, the body, would protect the gamete until it could be passed to the next generation. And that was really a milestone in life because it was the beginning of death. Life cared for the gamete. It was like bacteria. Bacteria just divide and divide and divide. They don't die. There's no such thing, really, as aging or death in a bacteria. There's no corpse. It just keeps going and going and going. Well, we do that too, to some extent, but only with our gametes. We have one lucky cell, if we're lucky and we mate. One sperm and one egg will continue on into the next generation. The rest of us becomes expendable. The job for the rest of us is to see that that one cell gets passed along. And that is what nature cares about. Nature cares that we do remain immortal, but not all of us. Just that one cell. The rest of us, the soma, is there strictly to see to nature's purpose of passing on that gamete. And then we become expendable. And that is why we die. That is why we age. And that is what nature cares about. She doesn't care that we live a long and healthy life. We just have to live a long enough and healthy enough life to be able to fulfill her purpose. This is really dramatically illustrated by many characteristics that we have that illustrate a phenomenon in aging called antagonistic pleiotropy, where there are many traits that we evolved with that are beneficial in allowing us to reproduce, allowing us to have viable children, but then ultimately end up killing us. Insulin resistance is perhaps one. Insulin resistance actually keeps us alive long enough. It protects the inside of the cell from glucose toxicity and glycation. But ultimately can give us cancer and heart disease. Nature doesn't care about that because most of the time it occurs post-reproductively. Now our lifestyle is so bad that now it is even occurring pre-reproductively, enough for perhaps nature to do something about it. There's no viable way for longevity genes to actually be selected for, because the power of natural selection wanes as we age. We don't have as many children when we're 70. And so even if there was a trait that allowed us to be healthy and strong late in life, it has nowhere to go. It'll just die with us. The traits that get passed along are those that allow for greater reproductive success. Be damned with living a long life. That's why we age. Then there's the other half of aging, you might say, of how we age. Now, how we age can be divided into kind of a competition, a balancing act between the amount of damage we accrue and the amount of damage we can repair. If we could repair damage as fast as it occurred, we would live forever. Unfortunately, the mechanisms that we have to repair damage also become damaged. Ultimately, damage will accrue. But nature's purpose is to pass on our genome to the next generation. And so there will be an allocation of energy. Since energy is not, in our ancestral history, was not infinite, and because nature has certain priorities, she has to decide if there are enough nutrients available to have children now, or if not, to delay reproduction and keep the soma alive. So if there is a nutrient deficit, we evolved with nature and life learning that you shouldn't even try to make babies now. There's not enough nutrients. All mothers know it takes energy to make babies. And if there aren't enough nutrients, then what life will do, it will divest its resources towards maintenance and repair so that the soma can stay healthy, so that it can perhaps reproduce at a future, more nutritionally opportune time. If there are enough nutrients available, then it says, make hay while the getting's good. And then the soma has done its purpose. And then nature stops caring for the soma. It doesn't try and kill you. It just doesn't try and protect you anymore. And so we ultimately then fade away, with the exception, hopefully, of one of our gametes. And so that allocation of energy towards maintenance, repair, or replication or reproduction is really important, and it's based on nutrient availability, and that means there has to be sensors that can detect what the nutrient availabilities are. And we have them. We've had them since the beginning of life. And they will regulate genetic expression. They regulate our genes. We have a fixed number of genes. All life has pretty much the same genes. The importance of genetic expression versus our genes, I think, is really illustrated by the little worm, the little C. elegans. A lot of research is done in aging on little C. elegans, little 2-millimeter worm. You can barely see it. Has 959 cells. Doesn't have any more than that. Really tiny. And how many genes do you think it has? 21,000. How many genes do we have? About 20,000. Has the same genes that we do. Obviously, it's different, and it uses those genes differently. But the difference is not just between a diabetic and a non-diabetic, or a heart disease patient and a non-heart disease patient, has much less to do with your genetic inheritance of genes, but has everything to do with how those genes are being played. Genetic expression. And what controls genetic expression more than anything are these nutrient sensors, because they control how long we live, whether we're going to repair ourselves or not, whether we're going to replicate or not. And over half of our genome is going to be expressed differently depending on what you eat for breakfast, depending on how you feed these nutrient sensors. The first nutrient sensor that we knew about was insulin. And I won't go into the long story there. It's really a nice story. I don't have the time on how it was discovered because I am actually quite familiar with Tom Johnson at University of Colorado, who discovered the first so-called aging gene, named it the age-1 gene, later found to be connected to the insulin pathway by Cynthia Kenyon and Daniel Ruvkin at MIT. Cynthia Kenyon was at University of California, San Francisco. And they then went on to elaborate on this insulin pathway that regulated aging in this little worm, C. elegans. Later, other nutrient sensors were discovered that affected aging, such as leptin, and more importantly now, probably TOR, targeted rapamycin. And that's what we'll be talking about. Now, we will go on, and I will give you an answer first. I don't know why that's black, so hopefully you can read it. It wasn't black on my computer. Your health and your lifespan is going to be determined by the proportion of fat versus sugar you burn over a lifetime. And that has to do with how you're affecting the genetic expression by nutrient sensors. You can just ask this question. Is it helping you burn fat, or is it making you burn sugar? Depending on what your answer is, will tell you whether this plan or supplement or routine that a person goes through, meal, is going to be healthy or not. Whether you burn fat or sugar is going to be determined by the communication of nutrient sensors. You should eat today to control the sensors that will tell you what you will need to eat tomorrow. Your cells will burn sugar, they will burn fat, depending on what the nutrient sensors have told your genes to do. So again, it's not just calories in, calories out. It's calories in, change your nutrient sensors, change your genetic expression, change your health. That's it. If you get nothing else, if you just remember that, you'll come a long way. Now, I mentioned how important it is, as far as I'm concerned, to go back much further than Paleolithic times. Paleolithic times really tells me pretty much nothing. But when you go back to how cells evolved from single cell into multicellularity in the so-called Proterozoic era, or eon they call it, 2.5 billion years ago, then you'll get somewhere. And I just want to illustrate the difference in time. And so prior to going through a ton of studies where things get a lot more complicated and I talk a lot faster, you get a little rest right now. I'm going to go back. At the very start of that, there was a red flash. And I want you to pay attention now. That red flash is Paleoman. Okay. We're going backwards in evolution. I actually reversed this. I have to give the person credit who actually did this. I reversed it, and I can't remember his name. But anyway, got it off of YouTube and just reversed it. Okay, so here we go. I don't know if you'll be able to see it on that board, but at the very beginning, you will say a flash of red, and that's Paleolithic man. Do you see it?
Yeah.
Okay. Now we go back. We can pay homage to all the life that has died so that we can live. And this is done in true relative time. There's nothing different, really, about how life worked here 400 million years ago as 2 million years ago, or as today. The rules of health and nutrition were laid down long before that. Now, in real time, I was going to talk about the evolution while these were playing around. These are the little cyanobacteria that also arose from protobacteria. This would go on for 3 minutes. Over half of our ancestral life, this is what we were. And the transition between that and colonialization and multicellularity is when the rules of health were laid down. It's when nutrient sensors evolved. That's when TOR, mTOR evolved. It's the oldest nutrient sensor we know, found in all life. Maybe even bacteria, we're not sure, but certainly all life beyond bacteria. It was so important to sense nutrient and energy availability that these nutrient sensors dictated everything. It dictated the biological imperatives. There's only 2. I do a lot of work in India. Very beautiful spiritual country. But from a biological sense, there's only 2 imperatives, as to eat and to reproduce. And that's what biology cares about. And the rules to do that were laid down there. Now we're going to go into cancer. Why? Because you just saw what cancer was. Cancer was our ancestors. We have ancestors whose prerogative it was and whose endeavor it was to divide as fast as possible. That's how it stayed in the evolutionary game. If you could outpace your neighbors, you would be selected. You would be left. There was a lot of food available. They could do that. Life was simple. Just multiply as fast as you can. I mean, they made rabbits look celibate. Yeah. This is what they did. Now, there's two major schools, you might say, of cancer research and cancer thought over the last century. Predominantly right now, cancer is thought of as a genetic disease, that we accumulate mutations, usually 2 or 3 mutations, that ultimately allows us, or forces us, to get cancer. That's the prevailing thought. Almost all the research money that's gone into cancer in our war against cancer over the last 50 years has been directed towards this type of paradigm. Have we made any progress? No. We've made some progress in diagnosis, so if you can diagnose it earlier, you can just cut it out, but as far as actually treating and curing cancer, we've gone nowhere. Maybe it's because they're asking the wrong questions, but we'll go on. This is a study that shows that in acute myeloid leukemia, just one type of leukemia, they analyzed the genetics of it, and they found there were 11 groups of genetics. So there were at least 11, they said, at least 11 kinds of AML, if one goes by the genetics. But then they actually went on to say that most patients had a unique combination of genetic changes driving their leukemia. In other words, each patient had a different set of mutations. So it's not surprising they haven't gotten very far in the genetic paradigm. Here's even worse. And this was just from a few weeks ago. A new study led by Cedars-Sinai investigators dramatically illustrates the complexity of cancer by identifying more than 2,000 genetic mutations in tissue samples of just esophageal tumors. 2,000 mutations. Now, if you go by the paradigm that it takes at least 2 or 3 mutations to develop cancer, and here in this one cancer you've got 2,000 genetic mutations. If you look at the different permutations, you have what amounts to a unique genetic genotype of cancer for every person on Earth. In other words, every person on Earth, if they had cancer, would have a unique genotype of their cancer. But even worse, yes, it gets worse than that, that different areas of the same tumor have different genetics. You can't talk about the genetics of a tumor. It's like talking about what your telomere length is. There's no such thing. If you're talking about telomere length of what? Your white cells, which is what they measure, your heart, your kidney, your nerves, they all have different telomere lengths and a different rate of attrition. Some cells hardly turn over at all, so they don't need telomeres. Then you have telomerase, which can regenerate, and any cells that need to keep regenerating, like our gametes, like white blood cells, have telomerase, and like cancer, they just add to the telomere length. So when you talk about telomeres, and I'll just mention this real quickly, and they're measuring white cells, all it's really measuring is the rate of turnover of white blood cells. It's a very expensive marker of inflammation, is what it amounts to. Now the other group says cancer is a metabolic disease. Well, yes, and I will agree with that, but it tells you nothing. It's like saying illness is caused from being sick. Everything is metabolic. Metabolism controls the genes. When life first formed, it was probably metabolic first and genes second. Genes make the metabolic sensors that then control the genetic expression. It's like saying, what's more important, your heart or your lungs? They feed into each other. You can't really separate the two. So this tells you nothing. Yes, it is a metabolic disease, but so is every disease, especially chronic disease. There's nothing that's outside of metabolism. Metabolism is the chemistry that turns food into life. Metabolism, most people now believe that metabolism came first when life first formed in the oceans 3.8 billion years ago. I started talking about metabolism of cancer a long time ago. This was a talk in 1999. It talked about a finding. This was done in Italy, actually, and they actually used pasta. It was the intake of macronutrients and the risk of breast cancer. The risk of breast cancer decreased with increasing total fat intake. Now, this is, again, 1999. This is when nobody would believe such a thing. The risk of cancer increased with increasing intake of available carbohydrates. Starch was the chief contributor and a positive association. The intake of saturated fatty acids, protein, and fiber were not significantly associated with breast cancer risk. And that was in 1996. Otto Warburg. How many people here have heard of Otto Warburg? He's gotten a lot of publicity lately. People today are resurrecting his ideas of 1926. He got the Nobel Prize not for his work with cancer, for something else. He was a very smart biochemist. A lot of people worked in his lab that are actually better known than him. Hans Krebs, Krebs cycle, worked in his lab, who, by the way, totally disagreed with him about most everything. And actually, Hans Krebs was correct. What Otto Warburg found was that in cancer, there was a predominance of lactic acid, a lot of lactic acid, whether there was oxygen around or not. A lot of people kind of misinterpret what he had said. He wasn't saying that cancer burned a lot of glucose. That's not the Warburg effect. The Warburg effect is that cancer burns a lot of glucose whether there's oxygen available or not. So that was what he discovered, and it's true. Cancer does use a lot of glucose. It's rapidly dividing. It needs a lot of fuel. But his major premise, his major theory, and this is what people are bringing up today, is that the reason that cancer relied on so-called aerobic glycolysis, it's glycolysis, burning glucose using glycolysis, even though oxygen is present, because typically then they use respiration in the mitochondria, he said they had mitochondrial damage. He said mitochondrial damage is the cause of cancer, and it forced cells to use glycolysis. In fact, he was very vehement his entire life for the next 3 or 4 decades. He was very vehement, said that you cannot have a viable theory of cancer unless you blame it on mitochondria. He called them grana. Well, you can't read that, so I'll have to read it to you. I don't know why the background is black on some of these. The most useful piece of learning for the uses of life is to unlearn what is untrue. By Ancestral, ancient Greek. More modern rephrasing of that is our ability to open the future will depend not on how well we learn, but on how well we are able to unlearn. That's Alan Kay. And that's what I'm going to be asking you to do. I'm even going to be asking you to unlearn what you haven't even learned yet. For those of you that aren't familiar with Otto Warburg, you're going to be hearing a lot about him, and most of what you will be hearing about from others will be wrong. I will tell you right now that cancer is not a mitochondrial disease. In fact, it is virtually impossible. I can't go into all the reasons, but I'll show you some. I talked about hyperinsulinemia. I will tell you right now that it's not fuel availability that drives cancer. It is going to be the genetic expression of nutrient sensors, especially those sensors that are very much involved in growth. And so way back in 1995, I talked about hyperinsulinemia potentially leading to cancer, along with all sorts of other diseases like cardiovascular disease, hypertension, stroke, diabetes, obesity, autoimmune disorders, mental disease and decline. I was a big insulin guy, maybe the first. That insulin was the root of all evil. And it's a big root, no question about it. Still is. Insulin and its metabolic effects a few years later. Insulin increases cellular proliferation. What does it do to cancer? It increases it. Later on, teleclinic with Joe Mercola talked about aging. Two of the major factors that will determine whether a person has cancer are the amount of sugar and insulin. Sugar is just listening to orders. It's what pushes sugar. It's listening to orders from insulin and leptin. You get insulin and leptin right, your sugar is going to be right. However, the converse is not necessarily true. You can bring down sugar with drugs, which is how diabetes is being treated. Almost all treatments for diabetes raises insulin. Not only makes the root of the disease worse, but it increases risk of cancer. And we know now that diabetics have a much higher risk of cancer. It's not necessarily due to their disease, it's due to their treatment. It's not just glucose. That's a major message I want to get across today. It is not just glucose. Glucose is listening to orders. Glucose does do damage, no question about it. It causes advanced glycated end products, all that stuff. I talked about that decades ago. But it is not just glucose that drives cancer. Okay? It is not aerobic glycolysis or even anaerobic glycolysis. Metabolic reprogramming in cancer. Here's one reason. We know that increased glutaminolysis is recognized as a key feature of the metabolic profile of cancer cells. We discussed the roles of glutamine by contributing to the core metabolism of proliferating cells by supporting energy production and biosynthesis. The cells coordinate glucose and glutamine as nutrient sources. So we know, and it's well known really at this point, that cancer likes to utilize glutamine just as much as glucose. Now, if that's the case, how can they have damaged mitochondria? How can mitochondria be driving cells to glucose? Because, you'll see in a later slide, glutamine is metabolized in mitochondria, not in the cytosol. It virtually can't be a mitochondrial disease from that, but there are many, many reasons that point to the fact that it's impossible for it to be a mitochondrial disease. Lactate is like sugar for candy cells, or for cancer cells. Okay. That's also well known. Cancer cells utilize lactate for many reasons. They utilize lactate even to ingratiate itself into tissue. The lactic acid actually helps dissolve some of the cellular matrix so that it can push itself and establish residence, so to speak. Lactate metabolism is associated with mammalian mitochondria. Taken together, our results demonstrate a link between lactate metabolism and mitochondria. Mitochondria actually burns lactate. Again, cancer is not driven by mitochondrial dysfunction. So all these people, there's a lot of money going on right now in the so-called metabolic camp that is researching ways to improve mitochondrial function to treat cancer. They will make it worse. They're wasting their money. We've wasted a century researching the wrong things in cancer, and we're continuing to do it now with the metabolic camp, which is looking in the wrong direction. And I'm trying to change that. Thomas Seyfried is the biggest one who's written a book. It's a good book. Travis Christofferson is an excellent writer who researched some of the stuff on Ida Warburg and wrote a book called Tripping Over the Truth. I've talked to him quite extensively. A really good guy and a really excellent writer. Just happens to be wrong. Same about Thomas Seyfried. Ketone body utilization drives tumor growth and metastases. Cancer burns ketones. So trying to put them on a so-called ketogenic diet, because thinking that ketones will inhibit cancer, and they're actually feeding them ketones. That's part of the therapy that Seyfried and D'Agostino are using right now. They're feeding cancer patients ketones, thinking that's good. It will be good probably for glioblastoma. For some reason, glioblastoma does not like ketones. But you can't translate that to the other cancers, because most cancers actually really do like ketones. You don't want to feed them extra ketones. Hepatocellular carcinoma redirects to ketolysis for progression under nutrient deprivation stress. So, if you are trying to starve cancer of nutrients, which is kind of the metabolic camp's mantra, is don't feed it, and I agree with that. Don't feed it any more than you have to. But nutrient-deprived hepatocellular carcinoma cells employ ketone bodies for energy supply and cancer progression. So feeding them ketones is not going to help you. And of course, there's a lot of hepatocellular carcinoma, not the least of which is by metastasis. There's a lot of liver cancer out there. Much research has examined the use of glucose and glutamine by tumor cells. Many cancers instead prefer to metabolize fat. Breast cancer. Breast has a lot of fat. Waste not, want not. Cancer cells will burn fat. Pancreatic cancer. You've got the omentum, nice supply of fat. Pancreatic cancer loves to burn fat. In other words, cancer will burn anything. It'll burn glucose, lactic acid, ketones, fat, glutamine. It's not a problem of nutrients. You're not going to starve cancer of its nutrients. I will tell you that. Can't happen. We review findings that suggest that age-dependent mitochondrial dysfunction is not sufficient to limit lifespan. Furthermore, mitochondrial reactive oxygen species are not always deleterious and can even stimulate pro-longevity pathways. In the last few years, that's been found pretty conclusively, that you actually need oxidation. You need free radicals in the mitochondria to stimulate pro-longevity pathways, to stimulate the repair, the genetic expression of repair. And when you take antioxidants, and they kind of poo-pooed these studies a long time ago, saying, it couldn't be true, that taking antioxidants will actually increase your risk of cancer. They found that epidemiologically, and I agree. I don't agree with most epidemiologic data. It's kind of hard to really derive anything from it because you really can't control all the things you would really need to control in an epidemiologic study. But they found a long time ago there was an association between taking antioxidants and an increased risk of cancer. Now they're finding very good biochemical proof of why that is. And there was I should mention real quick why I put this in, because the mitochondrial free radical theory of aging was a big theory a number of years ago. And that's where a lot of people now are saying, well, Warburg was right, that cancer must be a mitochondrial disease. Well, the mitochondrial free radical theory of aging has been really, you can't say totally disproven, but pretty much shunned. There's a lot of evidence against it. That, in fact, I'll give you some. The next one, there's a mutator mouse. In these mice, the proofreading function of mitochondrial DNA polymerase is defective. So they made a mutated mouse that can't repair the mitochondrial DNA. These mice are born with a mutation burden 30 times higher than that of aged wild-type mice. So they're born with 30 times more mitochondrial mutations than aged wild-type mice. And yet they have nothing wrong with them. Typically they're fine and they live just as long. There's many reasons for that. Cells can get rid of their damaged mitochondria. They can take good mitochondria from their neighbors. The good mitochondria will reproduce, and you get rid of the bad mitochondria. And mitophagy, TOR does that. We'll talk about that. So damage to mitochondria is not really a major cause of aging, nor can it be then a major cause of cancer. What about growth hormone? Now we're getting somewhere. We talked about that yesterday. Growth hormone is definitely valid. I have a friend, Andre Bartke, won the first Methuselah Prize, which is given the biggest prize in aging research, given to whoever can keep a mouse alive the longest. He kept a mouse alive at the time 4 years. Typically, the lifespan was 2 years. He doubled their lifespan by mutating growth hormone receptors. Mice with pituitary gland devoid of growth hormone-producing cells exhibit a markedly extended lifespan, as do genetically engineered mice with a targeted disruption of the growth hormone receptor, which results in low concentrations of plasma IGF-1. That was by Andre Bartke. That's the year he got the Methuselah Prize. The role of insulin and IGF-1. The main thing I want you to get out of this, I'm going to have to start going a little bit faster, is that insulin and IGF initiate their actions via homologous signaling systems. So many of the detrimental effects one hears about insulin is really due to its stimulation of IGF receptors. Okay? IGF, insulin-like growth factor, is called insulin-like growth factor because it is a cousin of insulin. In the little worms where a lot of these aging studies are done, and a lot of the so-called pro-aging effects of insulin were discovered, one has to understand that insulin and IGF are one and the same in these ancient organisms. Later, it was kind of a division of labor between the anabolic functions of insulin and the metabolic functions. Whereas the anabolic functions kind of turned into IGF, and the metabolic functions were handled by insulin. But they cross-react with each other's receptors. So IGF can stimulate insulin receptors and drop blood sugar, and insulin, especially in hyperinsulinemic people, which is everybody almost in the world, you will have significant stimulation of IGF receptors and initiate lots of growth, which can lead to cancer. And then very strong similarities exist between insulin and IGF signaling systems in yeast, worms, flies, mammals, humans. In other words, these nutrient sensors are pretty much the same throughout the history of evolution. So yes, you can extrapolate what happens in a worm to what happens in us. In fact, you can use human insulin in a worm, and it will respond just as if it was its own. It'll do the same thing in a worm that it does in us. Here's the same picture I think you showed. Yeah, and I only put this in there because I just wanted people to know this is called Laron syndrome. So these Ecuadorian dwarfs have a syndrome called Laron syndrome where they are remarkably free of cancer and diabetes, and they're born with low IGF. And I think you talked about that yesterday. So one of the explanations is it says it could be because cells must invest energy in either trying to grow and reproduce or on protection. In nature, dwarf models live longer. Ponies live longer than horses. Small dogs live longer than large dogs. It's a very fascinating field in aging. I want to mention, though, it's not so easy when you talk about between species. That's within a species. So within a species, smaller dogs will live longer than larger dogs. Between species, elephants will live longer than mice. And there's reasons for that. I won't have time to go into that. But within a species, definitely IGF levels are associated with longevity. So those people taking growth hormone as a anti-aging modality are in for a surprise when they die of cancer. They might look good for a short period of time, but it is absolutely not an anti-aging hormone. It is definitely a pro-aging hormone. Ungrowth hormone increases longevity, researchers find. Long-term effects of calorie or protein restriction. Our data provide evidence that protein intake is a key determinant of circulating IGF-1 levels in humans. and suggests that reduced protein intake may become an important component of anti-cancer and anti-aging dietary interventions. Okay? And this is absolutely true, not just because of IGF. Protein is the main driver of IGF, but there's even more important drivers. Insulin analogs and cancer risk. All those people that taking a bunch of, diabetics are told to take as much insulin as you need to control your blood sugar. Basically, eat whatever you want, just take enough, set your pump to pump you full of enough insulin to keep your sugars down. Well, there's a huge difference in using 100 units of insulin a day versus using 20 units of insulin a day. Even if they had the same, blood glucose levels. In fact, I don't know if people here are familiar with the Accord study that showed that it was a big study. I don't really have a lot of time to talk about it. It was actually broken by the New York Times that.
45 minutes.
Is it really? Darn. Okay. We have a long way to go. Those people who took more drugs, even though their sugars were down, ended up dying sooner, basically. Leptin. Central nervous system control of food intake and body weight, that's leptin. Here, this study showed a more critical role for leptin than insulin, both in controlling sugar, by the way, and fat. And chronically elevated leptin increases hypothalamic leptin receptor expression, causes leptin resistance, the disconnect between your body and your brain. Leptin regulates pro-inflammatory immune responses. We know that leptin levels are strongly correlated with MS. The link between high leptin concentrations and telomere shortening was even stronger than the link with obesity. Okay, again, the telomere shortening is just really a surrogate for inflammation, and leptin levels, high leptin levels, causes that inflammation. Obesity linked to aggressive prostate cancer. Leptin and IGF-1. Leptin's failure may be considered for the metabolic decline seen with aging. Long life was predicted by low levels of leptin. Amino acids are what regulate leptin and glucose, not fat. So the major driver of leptin secretion is not just how fat you are, which is what is commonly thought, but if you eat a diet high in protein and glucose, you will cause a surge in leptin, and these spikes in leptin are what cause leptin resistance. And this is still something that is not being appreciated, unfortunately. Protein increases IGF-1. High protein accelerates aging. Reduced protein extends life. Why TOR? TOR is really at the center of it. TOR was a pathway that was discovered when, in the 1970s, they gathered up a soil sample from Rapa Nui Island in the Easter Islands. It was a group of scientists, and they went to this remote island financed by a drug company to see if they could find something. And later on, it took 20 years, and they found that this soil sample contained something, a chemical that they called rapamycin, that was an antifungal agent. That's really as far as it went. So, okay, we found an antifungal agent, not that big a deal, but we can use it. Let's see how it works. And then they found out that it worked by inhibiting a pathway that they called the target of rapamycin, which they found also. They found that the rapamycin was an immune suppressant, and they had it approved to use it for organ transplant patients, especially kidney transplant patients. But then they found that those kidney transplant patients who were having their immune system suppressed from rapamycin also ended up having a lower incidence of cancer. Well, that raised some eyebrows. And they looked at this pathway a little bit more, and lo and behold, they discovered that this could be, and I would say it probably is, the most important biological pathway in the body. It is the switchboard. All of the nutrient sensors and all the nutrients feed into mTOR. Let's There it is. Okay? It's an enzyme complex. It's not a hormone. And do I have a pointer by any chance?
Yeah. Push
The red button?
Yeah.
Okay. All right, so you don't have to know all of this, but here is TOR, TORC1. There's actually two complexes, TORC1 and TORC2. TORC2, when it's suppressed, is what reduces the immune system. TORC1 is suppressed, well, it's actually stimulated by amino acids and glucose and insulin and IGF-1 and all other growth factors and AMPK and AMP to ATP ratio, and every nutrient sensor that there is, and nutrients themselves will feed into TOR. And TOR then makes the decision of what to do. It's kind of the CEO of metabolism. And it will affect genetic expression and tell that organism, tell that cell, whether it should grow bigger, whether it should divide, or whether it needs to hunker down and repair itself so that it can live a long enough life to be able to replicate at a future, more nutritionally opportune time. Everything goes into TOR. The key here is that TOR is ancient. It predated insulin by 500 million years at least. So insulin feeds into TOR. But because it predated even multicellularity, there weren't hormones around. Hormones are to coordinate the activity of 15 trillion cells or what have you. Okay? It, in single-cell organisms, it needed to sense the actual nutrients themselves. Amino acids, glucose. It goes straight to TOR. Don't pass go. They don't even have to stimulate other nutrient sensors. However, the most important, and I'll just keep going, this showed that the targeted rapamycin controls cell growth and therefore is, including IGF-1, it is upregulated in 70% of all tumors, maybe more. In other words, TOR drives tumor growth. Tumors upregulate TOR themselves. They can't really grow without TOR. The TOR pathway is important in many types of cancer. Yes, that pathway activation is one of the most common molecular alterations in human cancer. TOR inhibits autophagy. Autophagy is where, very important process of repair, is where we basically eat our damaged junk. Again, that's controlled by TOR. It's a way, so if you are under nutrient deprivation, what your body will do is determine what is expendable, including do you have damaged mitochondria. It'll eat your damaged mitochondria, that's a good thing. It'll eat your damaged proteins, that's a good thing, and use the components to make new and better parts. You can't be healthy without autophagy. We find that this is, if you inhibit autophagy and do everything else right in calorie-restricted animals, they will not live long. In other words, it is necessary. It is a necessary ingredient for almost any longevity pathway. TOR controls fasting-induced ketogenesis. If you talk about ketogenic diets, you're going through TOR. If you're going to make ketones, you're going to have to lower TOR. We found that a major pathway by which amino acids control TOR signaling is distinct from that of insulin. The key here, TOR is activated by free amino acids. The thing I'm going to have to go pretty quickly through these now, but I wanted to mention that the most powerful signal that raises TOR by a long shot are amino acids. So if you want to raise TOR and stimulate cellular growth and basically turn off maintenance and repair, the genetic expression of maintenance and repair, you would eat excess protein. TOR is the biggest argument against eating a high-protein diet because you will raise TOR. There's nothing else that you can do that they found that will lower TOR if you eat excess protein. You can lower insulin genetically, dietarily. You can do whatever you want. You will raise TOR if you eat excess amino acids. And there are some amino acids will raise it more than others. Lysine, okay, is one of the biggest. Yes? It's found a little bit more in animal protein than plant protein. That just means you could eat a little bit more plant protein than you could animal protein. For the same reason that animal protein is actually more useful, it becomes more detrimental when it comes to TOR. Now, something I just want to get in, because I know we're running a little bit out of time, but I do want to mention that an example, I talked about antagonistic pleiotropy, where those traits that might have been beneficial when we're young become detrimental as we're older. TOR is a major one. We need TOR. You cannot develop. They found that if you inhibit TOR during the developmental period, embryogenesis, you will not develop. It's lethal. You can't have low TOR when you're young. You won't grow. They're also finding out that you can take rapamycin, they fed rapamycin to mice, in middle age, and it will be as effective, if not more effective, than if you started younger in extending lifespan. So you don't have to worry about actually limiting protein when you're a young athlete. You're fine. What you want to do is limit the protein after you've reached kind of mid-reproductive years, maybe in the mid-30s. Your lifespan will be extended, and you will be just as healthy if you limit TOR in midlife than if you started earlier. So that kind of takes into account this antagonistic pleiotropy. That's why you can't just use what is good for nature. You can't just say, do what's natural and it's be good for you. You actually have to understand nature and then apply nature's principles to our own doing. And maybe we don't really like what nature has in store for us. And so we can actually fight nature, but it has to be done appropriately. Find out what nature did to keep us alive and strong so that we could replicate in the first half of our life and keep it going then in the second half. And we know that we don't want to keep going with high TOR. That's when we want to start lowering it. Here they say, we find it intriguing that the last one, this is an important one. Longevity and health were optimized when protein was replaced with carbohydrate to limit compensatory feeding for protein and suppress protein intake. Our results suggest that longevity can be extended in ad libitum fed animals by manipulating the ratio of macronutrients to inhibit TOR activation. In other words, they didn't use fat as a variable here. They were comparing carbohydrate and protein. Between carbohydrates and protein, it was more important, much more important, to limit protein than carbohydrates. In fact, if they replaced the protein with carbohydrates, lifespan would be shortened. Excuse me, it would be lengthened. Yeah. So if you had less protein, despite higher carbs, you would still live longer because you would be suppressing TOR more, because protein is a more powerful stimulant of TOR than carbohydrates. Effective dietary protein restriction on glucose and insulin metabolism. I'm going to go to the end. The anti-Atkins diet extends lifespan, low protein diets. The best drug to reduce TOR signaling to slow aging and chronic diseases associated with aging is available. You don't need to come up with drugs. Just don't eat too much protein. What is that? Very early on, I recommended, a quarter century ago, the diet that I proposed to treat my diabetic patients and cardiovascular patients and any of the chronic diseases of aging was a very low carbohydrate, very moderate protein, and high fat diet. At the time, there were maybe a half a dozen people in the world, maybe less, that were even knew anything about or even were contemplating or using a very low carbohydrate diet. It was very new. And all of the other people that were advocating a low carbohydrate diet were at least condoning, if not trying to push, a high protein diet. My partners at EADS were pushing, they wrote a book called Protein Power. They were one of the first. We were partners. But I said, you shouldn't be feeding them protein. But my idea at the time is because I knew that protein also stimulated insulin. Insulin was a growth factor. It stimulated IGF. So that was my purpose for not wanting a high protein diet at the time. Nobody knew anything about mTOR at that time. But now we know mTOR is really powerful evidence that a high protein diet is not beneficial. But even in my practice, I found that a high protein diet would not be optimal for diabetics. It's very hard to control their blood sugars. Their blood sugar would just be raised with the gluconeogenesis from the protein. So at the time, I was recommending about 0.75 grams per kilo of lean mass per day. If you were healthy, I was recommending maybe a gram. Now I'd say you should go down to probably 0.6 and even 0.5 grams of protein per kilo of lean mass. You can go down lower than I thought previously. That's the only difference, is you can really go down on the protein and you will be just as strong. You're not going to be protein deficient. Now, there is a limit, of course, but the reason has to do with autophagy. You'll just eat up your own damaged proteins. That's a good thing. And so you'll still be getting more protein, but it'll be coming from your own junk by keeping TOR low. The protein content in breast milk is about 1 gram per 100 mLs. They have an intake of approximately 1 gram per kilo per day of protein in breast milk-fed babies. I can guarantee you there will not come another time in your life where you will need more protein than when you were an infant breastfeeding. The first fasting mimetic diet. Wrote a paper with Duke University over a mean follow-up comparing my diet to calorie restriction over a mean follow-up of about 3 months. Body weight decreased. We weren't really giving it for weight loss. Serum glucose decreased 8.3%. There was a 50% reduction in serum leptin, insulin, fasting triglycerides, triglyceride HDL ratio, free T3 decreased 7%, while TSH did not change significantly. I didn't write on there. Also, body temperature went down 1 to 2 degrees centigrade. We got exactly the same changes that one gets with calorie-restricted animals in the same amount. And there was no calorie restriction here. There was no fasting. People could eat as much as they wanted, as long as they ate the right diet, a high fat, they watched their protein. The only nutrient I've ever had people count were grams of protein. I never have had people count calories. I've never had people count the amount of carbohydrate. The less the better. The only nutrient for the last quarter century that I've had people count is how much protein should they have per day. And now I've gone down a little bit on it. I did, however, tell people and advise them that they should not eat for at least 3 hours prior to sleeping. That way they're not digesting food while they're sleeping, they sleep better. So ultimately that kind of leads to an 11 or 12 hour fast. If they, by the time they have breakfast, wake up in the morning, shower, whatever. So that usually does lead to a little bit of a fast every day. Your health and your lifespan will mostly be determined by the proportion of fat versus sugar you burn over a lifetime. But once again, and that will be determined by the communication of insulin, IGF, leptin, and especially mTOR. Cancer is not glucose driven, nor is it driven by mitochondrial dysfunction. It is driven by growth signals. In other words, you eat to regulate growth signals, not the nutrients that are available, but how the nutrients affect the growth signals, particularly TOR. Life is not in the parts. We're all made of the same junk, all made of the same stuff. It's what you do with the parts that determines your life and health, and that is largely determined by TOR levels. Okay, and I know there's a break right now, but if you want, I will be more than happy to take questions.
We're taking 2 questions.
Yes, sir.
How do you adjust protein intake for someone who's an athlete? You still go by the same 0.6?
Well, again, it depends on how old that athlete is. So if a young athlete, an Olympic athlete that might be 30 years old, they can eat more protein without a lot of damage. They can afford high TOR at that time. They're finding that there isn't a whole lot of advantage to limiting TOR until you're maybe 35, 40 years old. So if you're an older athlete, then you have to make up your mind what you want. Are you going for the Senior Olympics? And is the trophy important to you? In which case, you could eat more protein, but you might suffer from a shorter lifespan. One has to understand that athletic performance and health and longevity are not necessarily the same. There's a lot of great athletes that die young, and a lot of them know that they will. Even take performance-enhancing drugs and risk the consequences. But you can still, even if you limit your protein, I'm not particularly limited athletically. And I rarely get more than 45 or 50 grams of protein a day. Yes?
I'm sure you know the work of Dave Asprey, who recommends that 2 days a week that you eat really low, like essentially no protein. So it would average out over the week to something like what you're talking about. For 5 days a week, you'd eat closer to 0.75 to 1 gram per kilogram. What do you think of that?
I think there's probably a lot of pathways to lower TOR, and that would probably be one way that one could do it. I think that there, certainly room for days of fasting, and I think what we're showing is the biggest benefit of fasting is the limiting of the protein. Now, certainly you're going to get some benefit by limiting carbohydrates and lowering insulin and that sort of thing, but again, they all go through TOR. And the most powerful stimulant of TOR is protein. So if you just limit your protein on particular days, you're fine. Or you can limit your protein maybe 12 hours a day. Or you just have 1 or 2 meals of protein per day. A lot of people made a big deal of the Kitavans and why they live a long life. Well, they eat 1 protein meal a day. They don't live a long life, by the way. They had a lower incidence of diabetes and cardiovascular disease, but not a particularly long life. I wasn't exactly sure why everybody was making a big deal of them. Centenarianism was totally unknown. They're small people. They have a lot of advantages. The real question is, why don't they live a long life? And they did eat a lot of carbs. So carbs, there's certainly a place for lowering carbs. Don't get me wrong. I haven't changed that at all. Basically, what TOR has taught me is that what I was saying a quarter of a century ago, and that we have to watch our protein in addition to carbohydrates, is more true now than, well, I can't say it's more true, but we know it's more true now than it was then.
All right, one more and then we'll wrap up.
We'll get you. We'll get you both still.
Do you mind if I just all in the elderly who already have lower muscle mass and are more prone to, falling, getting hip fractures, which is increasingly mortality?
Right. There certainly was a study that was done that showed that limiting protein in 65-year-olds and older might shorten their lifespan. But you have to understand that these people are people who have probably eaten a high carbohydrate diet most of their life. And we know that insulin sensitivity declines as we age. So they become more and more insulin resistant. And what happens then is they become more and more gluconeogenic. And they tear down their lean mass. If you're eating a low carbohydrate diet, I have a feeling that a study in those people would be totally different. I don't think that they would be as gluconeogenic. I don't think they would be eating up their lean mass so much. Therefore, I don't think that they would be needing a higher intake of protein. What they're doing is they're trying to eat more protein to substitute for the lean mass that they're breaking down. The real key then is don't break down your lean mass, and then you don't need so much protein. So I don't really buy into that. I think that, in fact, it's even more important to limit your protein when you're elderly, as long as you're doing it properly and you're limiting your carbohydrates so that you reduce gluconeogenesis and you rely on fat burning. Again, remember both the first and the last slide. Your health and lifespan is to be determined by the proportion of fat versus sugar you burn over a lifetime. The reason people get osteoporosis is not because of a lack of calcium, but because they're burning up the protein in their bones because they're addicted to burning sugar and they're not burning fat. If you're burning fat appropriately, you're not going to be eating up your bones and muscle so much. I mean, you're still going to age. I can't say that this is a recipe for immortality, but we can stretch out youth, and we can increase our chances of being healthy and reduce our chances of getting cancer. Ultimately, we should all be dying of cancer. Hopefully it will be much later. We shouldn't be dying of heart disease. We shouldn't be dying of diabetes. Those are pretty much man-made diseases by our lifestyle. Cancer is what ultimately that would get us.
Maybe pneumonia at 100.
Yeah, maybe. Yeah, something like that. Yes?
Okay, so what about muscular atrophy as we age?
Muscle atrophy. Well,
Just to finish, we absorb bioavailability of protein decreases as we age, for one. And secondly, we do atrophy, especially if we're not active. So sure, you have to consider as a consideration as well, not just reducing protein. Because how do we maintain lean muscle mass if we're not moving?
well, the biggest variable, I think, in the loss of lean mass is that we end up burning it for sugar. And we end up burning it for sugar because we have hormones that tell us that we need to burn it for sugar. If we change our hormonal genetic expression so that we can burn fat as our primary fuel, we don't need very much glucose at all. In fact, you hear that your brain requires glucose. That is absolutely wrong. Your brain can work perfectly well with no more, maybe even less than 10% glucose. And that's been shown quite definitively. And you can get that 10% glucose from eating fat, from the glycerol. So if you eat a high-fat diet, all the glucose your brain would ever need, or any of your tissues, or your red blood, any of your tissues would need, would come from the glycerol of the fat plus the lactic acid and the ketones. And so your intake, the amount of gluconeogenesis that you have at that point goes way down.
I understand your point, but I'm sure we've all seen the cross-section of the thigh muscle where with a 75-year-old triathlete and a 45-year-old triathlete, and then somebody who's 70, and it's pretty much like hardly any muscle at all in a 70-year-old individual.
Yeah.
So that's obviously that's dietary agnostic, right there.
Well, no, it's not dietary agnostic.
Well,
They were probably eating a high-carbohydrate
they're
diet.
having a low-protein diet, that's for sure.
Well, maybe, not necessarily. They could be eating a high. If it's not. You don't gain mass. You don't gain mass just by, sitting on your couch and eating protein.
Right, but so is the other.
Hey, guys.
athlete. No, it's the athlete.
All right, thank you. One more question. Is it going to be a real question?
What are your
Good
thoughts
question.
of using metformin or rapamycin for brain
I think that rapamycin is a bit of an experiment. And I will tell you, just a little secret, for the last month I've been using it just to see. I haven't noticed, of course that's long term, but I haven't noticed anything adverse. And very low dose, and not every day. And they're still experimenting on how to use it. They said maybe 1 week on, 1 week off, so you don't suppress TORC2. TORC2 apparently is less sensitive to rapamycin than TORC1. And what you're trying to do is maintain your immune function. So you want to maintain kind of a higher TORC2 level while you lower TORC1. And they're kind of experimenting with various recipes of taking rapamycin for that. I'm playing it, really conservative. As far as metformin is concerned, I can't give you the reason why right now, although I will be writing it in my book. I'm writing a book on all this. I think it's the best drug out there as far as that goes. I do think it's very good, and I think it should be part of the repertoire of any cancer patient.